Hello YouTube, this is Sam from Tiger Schooling, and we are going to make a series of video on and about the enzymes. So this is the first lecture on enzyme, and we make it as easy as possible and as informative as possible. So be with me till all the lectures to understand the complete enzyme. So basically the word enzyme is made up of two words, that is an and zyme. So an is actually the word which means within, and zyme which means leaven, or you can say rise, which is a Greek word. So actually they are the protein in nature which have high molecular weight, protein in nature, except the ribozyme. So the main thing that enzymes are used for, actually, they are the biocatalysts. So if you know what is catalyst, so actually catalyst is actually an agent which increase velocity of reaction by reducing activation energy. We are coming to that idea about reducing the activation energy of the usage of the biocatalyst. But let's understand that this is a biocatalyst which is never used up. Even in the end of the reaction, you will find the catalyst which actually help to boost up the reaction, but never itself is used. So one thing more to add up in this enzyme, that it never initiate reaction, only it accelerate it. It fasten, or you can say it makes the reaction go fast. Now let's talk about the catalyst. Alright, so we have the catalyst. Enzyme is actually a biocatalyst. So the catalyst is actually, I've talked about, there are agents which increase velocity of reaction by reducing the activation energy. So what is activation energy? In this graph we can see, okay, I haven't mentioned the activation energy. Alright, I will just go through that one in just a bit. But remember, this is actually the minimum amount of energy that is required to activate atoms or molecules to a condition in which they undergo chemical reaction. In just take an example that you are just sitting with a friend, and somebody is kind of teasing you again and again. This is not obvious. I'm just correlating this. You're sitting around friends and somebody is kind of teasing you and making fun of you. You will bear that friend, for a few moments. You won't say anything, but there will be a limit. After that limit, you will say, no, stop. So what does that mean? You have crossed that limit. So this is also a limit. Like, to start a reaction, there's a limit, and that limit is activation energy. When you cross that limit, you go as up as high, and the reaction starts. So what is the catalyst? Agents which increase the velocity of a reaction by reducing the activation energy. So for example, this a reaction is happening. This is a reaction in progress. If you're not using a catalyst, you will need this amount of energy. Like, you will need that amount of energy to start a reaction. Like, as we go upward, the energy increases, and this is how the reaction progresses. But without a catalyst, you will need a lot of energy because the activation energy is much higher. But if you're going to use a catalyst, enzyme, which is a catalyst, which is a biocatalyst, if you're going to use the enzyme, it is going to reduce that amount of energy required to a minimum amount where the reaction can go easily as possible. In human body, it is very necessary, because if you're not using a catalyst, we need to raise the temperature to make a reaction go fast, to reach the activation energy. But we can't, our body temperature is constant, so we cannot raise the temperature. So what we have to do is, we have to use the enzymes for this reaction. Alright, few other things I have to discuss about the enzymes. Like, this is introductory, so I'm going to put up some few things other. Look at this picture. Consider this thing as an enzyme. This is an enzyme. Alright, these are our enzymes, right? And this is which is on the top. It's the substrate. So what is actually substrate? It's a compound on which the enzyme acts. So this enzyme is going to act on the substrate, to convert it into something new, product. We call that product. So this is a substrate which actually attaches to the enzyme, and finally which is converted into the product. This glucose is the final product. So the maltose come and attaches to the enzyme, And when on the active side, so one thing more, the active side where substrate and enzyme connect to each other. This is the active side where both they are attached. And another side which is called the allosteric side, which is not present on every enzyme, but on a lot of enzymes it is present. So what is allosteric side? So it's the other side than the active side, except there are a lot of time, activators, actually a lot of times inhibitors attached to this point, so which it changes the shape of the active side. We will be going to discuss that thing in later videos, but just to give an idea, inhibitors attached to this which changes the shape, and the enzyme activity is reduced, or sometimes it falls off. And one thing I have already talked about, the active side, which is region of enzyme where substrate bind. And one thing more I have to add up in this lecture today, it's the idea of hollow enzyme. Hollow enzyme. So what is hollow enzyme? Sometimes what happens that not only the enzyme is made of protein, but a lot of enzymes which have another part that is non-proteinous part, so which actually make it more stronger, more reactive than normal. So on the hollow enzyme is an enzyme which is made up of the protein part, protein plus non-protein part. So the protein part is called the apoenzyme, and the non-proteinous part is called the coenzyme. Don't get confused with the terminology of the coenzyme. I'm going to define the prostatic group, the coenzyme, and the cofactors in another video, and make them easy as possible, because most of the students get confused with the word coenzyme, cofactors, and the prostatic group. So the word coenzyme is actually in the biochemistry used in two senses. Like if you find the coenzyme sometimes in a heading, and you read a lot of prostatic groups in it, so in that case it means that it is something which is with the enzyme, like coenzyme, like coordinate, they are together with the enzyme. So it could be prostatic group, it could be the coenzyme, actual biochemical coenzyme, it could be cofactor. So get the idea of the coenzyme. So coenzyme, any non-proteinous part, you have to understand that. It could be the coenzyme in the biochemistry, we will coming to that soon. It could be prostatic group, it could be cofactor, any of that thing. All right, this is an English word meaning co, together with the enzyme. So it is a non-proteinous part in the case of the hollow enzyme. Few other terminologies we have what we call the what we call the monomeric enzyme. So we have other terminologies as well. We have got monomeric enzyme. So these are the enzyme which are only made up of one polypeptide chain. Monomeric enzyme, only one peptide chain. If you get the word oligomeric, it means it is made up of more than one. And there's one thing more, and this thing to add up, we have got, I'm going to add up that thing in here. It's called the multi-enzyme complex. So what is multi-enzyme complex? So what happens, like sometimes for a substrate, there are a few substrates that are not only one enzyme act on them. So few enzymes join together, combine together and make a complex of enzyme. Complex means something made up of a few other things. It could be the enzymes are made up of more than one. It's a complex of enzymes together. So it's actually stable. The assemblies, assembled together of more than one enzyme, which is called the multi-enzyme complex. So they complete becomes, if you break the multi-enzyme complex, they become completely inactive when you tear them apart. If there are a few examples in the case of the multi-enzyme complex, like all the enzymes come together, only that multi-enzyme complex is going to work. If you break them up, they're not going to work. So we have some example, that is the fatty acid synthase. It's and we have got prostaglandin synthase. So this is it for today, because I believe I have covered up a majority of the basic ideas regarding enzyme. So if you get the whole thing about the enzyme, I make sure you will be going to understand every other lecture. So our next video will be on the coenzymes, cofactors, and the prostatic group, and I make sure to make them as easy, as little confusing as possible. On the third lecture, we are going to discuss about the metallo and the metal activated enzyme, and finally we're going to classification, and we have other lectures as well. So thank you, keep visiting Tiger Schooling, and make sure to comment us and let us know what would you like to learn.